Ma'am, now we go ahead and invite Dr. Buddhadev Das, who is Chair, Professor and Chairman at MD Institute of Molecular Medicine and Stem Cell Research and Dean at Health and Allied Sciences. Dr. Das, he has made outstanding contributions in the field of cancer research, tumor virology, human genetics, and stem cell research. So during the last 48 years, he has done extremely distinguished work. He's, established himself as a molecular oncologist. He has also worked with, Nobel laureate Professor Haralzer Hossein at German Cancer Research Center. And he has pioneered a lot of significant work in India on human papilloma virus. His present areas of research are stem cell cancer, stem cell biology, molecular epidemiology, transcription regulation, gene editing, drug development. So, sir, we are honored to have you here. Welcome, sir. So, sir's talk is.
Good morning, all. I'm just sharing this slide in a minute.
So, sir's talk is cytogenetics, chromosome nomenclature, and karyotyping. So, the stage is all yours.
So can you share the screen, please?
Yeah, I'm just sharing.
Yes, sir.
Right, so am I audible? Hello?
Yes, sir. Yes, sir. We can hear you, sir.
Audible, sir.
Right, is visible?
Yes, sir. It is visible.
Okay. So thank you for inviting to have interactions with the audience for the talk that I have been assigned, that the cytogenetics or cell genetics and chromosome nomenclature and karyotyping. So if we go this is a molecular dissection of the genome, or in a nutshell, the life story. This is the book of life, that human body, which can be broken into the cells, the smallest unit of the body system, and that each cell contains, except germ cells, has the 2n is equal to 46 chromosomes, or diploid number or haploid number, and that is, the haploid number is 23, the haploid genome, and around 25 to 30,000 genes, 64 codons. You have already had lectures, so you are pre-exposed. 3 codons code for the 1 amino acid, then 4 nitrogen bases, and DNA double strand. So this is a book of life that can be divided into, simply to remember, 23 chapters of a book of the life, 30,000 sentences, and 64 words, and 4 letters. This makes the book of life, and that is the main master molecule is the DNA, so which directs all the functions of the body. If the DNA is abnormal, so your function is abnormal, or it is pathogenic. So what we see in the everyday or the clinics by the clinicians, that is nothing but the abnormal expression of the genes. In general, we can tell. So what is it what is genetics? So just a definition, I know everybody knows it, but then genetics is the study of inheritance of the human characteristics from parents to offspring. Simple, the traits like genes or genes, traits can be called as the genes. That is nothing but the part of the DNA that carries the message, and that is translated into protein. So as simple as eyes, eye color and the skin color, or the complex complex material, complex traits like susceptibility to disease and the hereditary disease and all. And cytogenetics is the study of your the chromosomal study. The study is the study of chromosomes and their inheritance and behavior during the cell divisions. So, and what is genomics? The study of all genes and their functions are called all genes inherited and encoded in on the two diploid genomes. And the genomics is the study of all proteins. That is, proteomics is the study of all proteins coded by the encoded by the whole genome is called proteomics. So these terminologies to understand, and just the genetics and the human disease, what is the contribution that they have immense benefits from that, the reliable early disease diagnosis and detection of the genetic defects, and the designing of the new drugs, understanding mechanisms of pathogenesis, development of modern therapeutics and whatnot. There are a huge number of things. So if you go to the chromosome, that is, this is prepared by the lymphocyte culture from the blood cells, the blood collection, and then you do the lymphocyte short-term culture, 48 hours or 72 hours. This is the 48, the chromosome, this is called metaphase spread, and this can be counted that every chromosome is in duplicate, means they are homologs. So it is a 46 chromosome, human chromosomes that can be counted. That is how it is counted. This is nearly, if you have to count, then you should have a very well spread chromosome. They are in a nice right side, you can see. So you did not go to other slide where this is the barking deer, left side barking deer chromosomes. That is only 6 chromosomes. How long and how beautifully it is demonstrated. And these are the different cycles. Using DRDU, you can go for the second cycle, third cycle, where they are, and where is the even if this the clean preparation with the simple metaphase plates, you can distinguish what is there any deletion, breaks, and any abnormalities in general. But you can go to the detail in the molecular level where you do the molecular the multicolor and the chromosome imaging. So that will come just in a minute. So haploid genome is 23, 2n is 46 are diploid, and 2n plus or 2n minus that goes to the aneuploid. Whether one chromosome is extra or less, that is, or more chromosomes are less or more, it is aneuploid more than 46. In case of human, but every species has the specific chromosome number, 2n number, that cannot be varied. Then that becomes a different individual or different organisms. Like if it is a 46 or 92, or it is a 40, then it becomes a mice chromosomes or the mice genome. So it cannot be. And then the polyploid, just the previous speaker has spoke about the 3n or 4n. So it becomes polyploid, multiple of the haploid. So 2n is equal to 46, 22 pair of autosome, 1 pair of sex chromosomes, and X that the different category of the chromosomes that you find, that you have the Mendelian pattern of the inheritance, that you get the zygote, the parents, the offsprings, the gametes are XX and XY, and then that is you get the 25% each, or 50% males and 50% females. That is you get the zygotes or the babies. That is you have the standard Mendelian pattern of inheritance. But then again, here the chromosome, different size of the chromosome. That is you have the metaphase chromosome that is shown. It is easily visible. So that's the metaphase chromosome is used. So it has the metaphase means the centromere, The middle is the centromere. From there, the two arms, the upper arm is called P arm, and this which is a short arm, and which is the bigger arm or the long arm is the Q. That is, you know all. So I did not go much of the details. And then you have the acrocentric chromosomes, the submetacentric chromosomes, and then acrocentric chromosomes and telocentric chromosomes. But that is based on the centromere. When you do the karyotypes, that is generally where you can see it here, the two sister chromatids joined by the kinetochore or the centromere, and then you have the short arm and P arm and the Q arm, and then there in their whole chromosome is having, we will come to the G banding, where you will know that the bands, it is shown here also, there is a dark band and the light band. So light band is called euchromatin, and the dark chromatin is that staining gym-sustained chromosome part is called the heterochromatin. These are called constitutive heterochromatin, and you will come to know facultative heterochromatin also in a minute. So, but it is most beautiful things is that the chromosomes, there are 46 chromosomes, they cannot merge together to become dicentric or any other different chromosome because they are protected by the telomeres, which has a cap. It is called as the cap. It is a cap on the chromosomes. That is, it is a repetitive sequence. It is the TTA and GG. You have no the hexanucleotide repetitions of this. So this is a repetitive DNA or heterochromatic DNA. So this cap doesn't allow to fuse with the other chromosomes. So it is protected the integrity of the chromosome stability, prevent chromosomal degradations and all this. So it is the genomic integrity is protected by this. This is, I will say a little bit more, that it is around 5,000 to 15,000 base pairs. But every cycles, beauty of that every cycles that some 50 to 100 base pairs are deleted when the cells replicate. The DNA, the two chromosomes, the one chromosome become two chromatids, and then one become replicated into the four chromatids and the two chromosomes. Ultimately, this and this is and during this each replication cycle, they lose the 100 to 50 to 100 base pairs, and at times comes. That's why it is related to the aging, longevity, and the cells, and ultimately they die. If you culture the cells for the 6, 7, or 10 passages, after their cells die out, because in the in vitro conditions, when you grow in the culture, they cannot grow beyond that if you do these the lymphocyte cultures. But the epithelial cultures are different. So if it is they have a telomerase, that is important, that it can replace this the hexanucleotide repeats, and if the telomerase is maintained in case of the cancer, in case of the stem cell, and also in gametes, this telomerase is present and they are overexpressed, then it can go to the carcinogenesis and so maintain the telomerase so that it is not aged. So that is a beauty of it. So this is briefly, and it is right side is shown the how the telomerase can be stained with the human karyotype with the telomeres by the image with the you can see with the this is the specific the fluorochrome used for the repetitive DNA sequence. That what beautifully it is seen that each chromatid is the tip is telomere staining is seen. So ISCN, the Denver Conference in 1960, it was initiated at that time. They said that the chromosome is a chroma means color and soma means body. It's a colored body. When you stain with the particular stain of the which stains the chromatin, taken by the chromatin, the correct chromosome number was decided at that time is the 46, not 48 earlier by Tizo and Levin in 1956. And these are some the catalog that the Ingram discovered. The gene, sickle cell anemia, Lejeune, the Down syndrome was chromosomal aberrations were detected in 1959 by Lejeune, and 1959 59 also the Ford and Jacobs Y chromosome in male sex determination, it was discovered, and P.C. Noel and Howard Hungerford discovered the lymphocyte culture. blood culture, simple short-term culture, and discovered the CML Philadelphia chromosomes, the 921 translocations. So the 922 translocations. The chromosomes can be divided in the 7 groups, A to G. A groups are three metacentric chromosome, 1, 2, 3 chromosomes, and B groups are 2 submetacentric, 4, 5, 7 is the 7 to 12, 6 to 12 are submetacentric. D group, their picture is given in the left side. D group comes as acrocentric, and the E group is the submetacentric, 16 to 18. F is the metacentric, G is the acrocentric, and the X chromosome and Y chromosome, XX or XY. So these are the karyotype, human chromosome showing the triple 18 chromosomes. That's the Edwards syndrome. So you can easily make out this G banding chromosome. You can pair the accurately by G banding. Then this is also G banding chromosome showing the trisomy 21. It is the Mongols or the Mongoloid, the Down syndrome. And then you have also this is trisomy. It is the long chromosomes. I have no time as to describe how the long chromosome can be prepared, and the banding can be easily distinguishable. So this is also Down syndrome picture, triple 21 trisomy. Then there is a spectral karyotyping represented with the multicolor. You can do wonders. Each chromosome they have the different color. Each chromosome pair has the different color. You can have more than 20 colors, and it is because of the human genome sequencing, and you can have the genome and the gene particular for that the chromosomes or that particular region or deletion or other defects. So that can be also tagged with the fluorochrome, and you can easily identify this can be karyotyped digitally. And now this is also schematic cytogenetic, so the spectral karyotyping. This is also spectral karyotyping, but all the defects can be detected. I have a less time to go ahead with that. This is also a chromosome picture, but this is, you can see that 17 translocations also, and the 15 translocations, and they are shown in the 2 places, and chromosome 1 and 12 translocations. So this could be possible by this multicolor system, spectral color or chromosome mapping, or the chromosome. Then this is also a comparative genomic hybridization. So this is also by you can find how the loss of gain that is translocated, and you can see that the 4 chromosomes and 10 chromosomes are involved. So this is Turner syndrome 1 chromosome. The beauty of this I am showing because these are my prepared by myself in the Maulana Azad Medical College. My initial days of career when I had with the collaboration of the gynecology department. These are my own photography on the karyotyping. This is also a case from the Saranpur. This is the Klinefelter syndrome 47XXY. So this is the buccal smear for the female showing the bar body. The nuclear membrane shows the presence of the bar body. The dark stain, you can say easily the bar body. So X activation, there are 2 X. So it is a doses of the genes are double here. But human, in the males it is XY, in the female it is XX. So nature has done that you cannot have the double dose of the gene doses in the female, then they will be super female. So nature has the inactivation. It has inactivation of it is XQ13 region is the X inactivity region. It is called X axis. And the key is in that initiate the inactivation, encodes the non-coding RNA and spreads all over the inactive X, undergoes epigenetic changes, loss of RNA polymerase II. So they are not transcriptionally active. Thus the inactive X loss chromatin modification and functional role. So this is the XY chromosome both involved in the sex determinations. So as I told, this X is region you can see in the Q arm and there is a fragile X also responsible for the mental retardation and many others. So this can be also located by the X chromosome. They are located, and it's important in the Y chromosome, the SRY, the sex determining factor or the X determining genes, TDF, and the azoospermia region also they are in the Q arm, AZF. So these are important things which can be easily done by the banding, and you can see that whether this is missing or absent or translocated. So this micrograph showing the fetal XY sex chromosomes, the Y chromatin and X chromatin bodies. So yellowy one is the X chromosome, and the pinkish one is the Y chromosome, where it is in the last one. You can see only the Y chromosome and the X chromosome. They are differentiated, can be differentiated with the color. So you can easily, not for the just for sex determination, but for the other molecular identifications. So it can be used for this. The TDF is the test determining factor. Just now I told SRY gene is the sex determining region, or the genes of the chromosomes, and the Y chromosome. XY is the deletion of the S or SRY gene. And if we find the XY females, it can be, you can go through the karyotyping and this rectal karyotyping, the deletion of the mostly SRY gene, and the XX males, which I have found also. I have shown that it is translocation the SRY gene, SRY gene to X chromosome. Bar body also the indication of this, how many X chromosomes are there. Barr and Bartram discovered this first time in 1949 in the nuclei of the neurons of the cats. And this is a formula is NX minus 1. That is sex chromatin body. That is if you have the two 60% will be showing the bar body, not 100%, but literally in the adult females. But if it is less than 5% or around 5% or 6%, then you can think of that is a primary amenorrhea and a lot of problems that see maybe a Turner syndrome or other problem. Bar bodies from oocytes and germ cell appear only, they are absent in the oocytes in the haploid genome of the germ cells, but they appear only at the 12th day of gestation in the embryo, and in the 16th day of cells in the embryo, and extraembryonic membrane in the 12th day. And these are random inactivation. We do not know which one is still, that it is a parental X or the mother sex, because 2X in the female. But in marsupial, it is known it is a parental X that gets inactivated. So inactive X is late replicating. It's a facultative heterodynamitization. In the G banding, you have seen throughout the chromosome dark band, white and light bands. That dark bands is there are interstitial heterochromatinization, whereas it is a whole chromosome is heterochromatic, means they are inactive. So this has no function. That's why it is called heterochromatinization of this facultative heterochromatization. Methylation, that is because of the methylation of the X genes promotes and maintain the stability of the inactive states. And so non-coding RNA and chromatin modifying complexes, they are on the detailed molecular biology involved in the dosage compensation. This is called as the dosage compensation in mammals, that the gene dosage is compensated by inactivating one of the X chromosomes. So 1940, Alfred Jost first described the mammalian sex determinations, and Bridges described the chromosomes in Drosophila, and Penter described the human X chromosome, X and Y chromosome. So all genes inactive in the undergo the inactivation. 50% of the genes always escape inactivation, and 10% genes have heterogeneous expression. So it is not totally inactive, but there are some functions also. In mammals, it is XX, XY, and this dosage compensation genes is achieved by transcriptionally repression or inactivation of the majority of the genes. That's why we call majority of genes located in one of the X chromosomes, and it is random. And that I have already told. Then the non-dejection is the most important thing to study. So it is the normal, I will not go into the detail. And this is the meiosis 1. You can have the non-disjunctions, and in the meiosis 2 and 1, anytime, any stage, you can have the non-disjunctions, so that it's a delay in the separation in the anaphase because of many of the reasons, the aging and many other factors influence. So I will not go into that because of the paucity of the time. So you can see that how that n plus and n plus, when it is n plus and or n minus, they are all aneuploids, but it is a normal one, n. So whatever it comes, it is after the NFS separation, either in the meiosis 1 and meiosis 2. If it is n plus or n minus chromosomes, then it is aneuploids. So this is a developmental differences in human genital duct. So it may not be just, I wanted to mention how that Mullerian ducts and Wolffian ducts present in the genital ridge. In males, Mullerian ducts degenerated under the influence of the AMH, the anti-Mullerian hormones, and secreted by the Sertoli cells or the of the testis, and then the Wolffian ducts and ducts differentiate into epididymis and differentiate into the seminal vesicle, and by androgen secreted by the Leydig cells. And in females, the Wolffian ducts regress and Mullerian ducts differentiate into oviduct, uterus, and upper vagina. So this is the how that the differentiation of the ducts occurs. But now comes the cytogenetics nomenclature, very important. The international system of human nomenclature, the cytogenetic nomenclature, serves as the principal reference of for describing human chromosomes complement, including the chromosome abnormalities. ISN was formed first by the Charles Ford in 1960 in Denver Conference, hence called the Denver Conference ISCN 1960. The later edition of the ISCN 2023 was published in 2012, and revision was done of the 2009. The ISCN is considered is the Bible of the human cytogenetic and serves as a guide to assist in describing or analyzing the cytogenetic and molecular cytogenetic defects on the abnormalities and the clinical cases. The ISN committee, comprised of the elected member of the experts of the human genetics experts representing each continent of the world, and meet periodically to discuss changes in the field, which are added, updated, and the ICN. This nomenclature must be followed for all human cytogenetic laboratory and cytogeneticists of the routine analysis and the report writing or reporting and the publications, including quality assessment and the lab accreditations. With the help of human chromosomes are identified across the size, position, and G-banding pattern, his P, Q, arm, and all these aspects are dealt there. So I need not to go much of the detail. Provides a list of symbols, abbreviation in terms of the set of rules. What are the abbreviations to be used in the chromosomes and abnormalities? So everything has to be followed by the ISCN pattern. What are the recent changes? On the covers of the ISCN booklet, now 2023 and in the 2020 edition, the chromosomes become first smaller, and then it becomes schematic diagram, and later on it's still, but it was there till 2016. But then 2020 edition was published with the front page not showing any chromosome anymore, and instead there are 14 vertical lanes, and each lane held by the 26 different color horizontal bars. The name of the International System of Human Cytogenetic Nomenclature, the abbreviation still remains as ISCN. So
I'm sorry, sir, but we are running a little short of time. A little humble request to please.
Yeah, 5 minutes.
Yes, sir, 5 minutes. 5 minutes.
I just completed that count. Just to it is main thing is over the what is to give the little bit of the molecular understanding that what is already been told my colleagues and others. What is a gene? A gene is a strand of DNA or RNA, which are the like eukaryotes, the viruses have the RNA genome, which carries the information to make the particular protein. But if the gene is abnormal, the protein is either absent or truncated, and it's function normally, and that creates the pathogenic condition. Discovery of the DNA already told that 1953, James Watson and Francis Crick and Rosalind Franklin and Maurice Wilkins. But James Watson and Francis got the Nobel Prize, and the Wilkins and Maurice Wilkins. But Rosalind Franklin, lady who died of the breast cancer, but she only discovered the X-ray diffraction results showing the double helix. And but then before she interpreted differently, but these three people interpreted and published the paper in Nature, and she was they got the Nobel Prize in 1962 or 68, I think 62 or 68, and the Rosalind died in the 1958, so she could not get any Nobel Prize. So that's the story many people do not know. Chemical structure, you know, ATGC, and this the DNA structure and the deoxyribose sugar. This is already dealt. But Hargobind Khorana, which I have the chairs of the Hargobind and Maulana Azad in Amity. So that's why I need to show his picture. He's our proud Indians and who got the Nobel Prize. Along with the Marshall Nirenberg, it is because of discovery of the genetic code. This is most important, the discovery of the DNA, but it is no less than discovery of the DNA. The triplet codons replace the amino acids that already been told by colleagues. So Dr. Viswas, that the 3 codons makes 1 amino acids. So these are the 64 codons. This was developed by first by Hargobind Khorana. And this is important, is the central dogma of life, that the DNA is double helix, but when it is transcribed to RNA, this is the only RNA which is mRNA, is the coding RNA. No other RNA. There are several RNAs, rRNA, tRNA. There are 7 RNAs. They are not coding. This is the only RNA, mRNA, that codings, that messenger RNA. It takes the message to code for protein. So transcribe into the single-stranded. These are the codes are also in different colors shown, and this is the single-stranded mRNA, and this mRNA can translate into protein. So it is shown that how the AUG become methionine, and like CCU and all, it is being shown how it is translated into proteins. So this is a central dogma, that then the proline is isoleucine, and the glycine and the serine, etc. But the main, how do you come into this world? It is all known to you. I don't see that, but important thing that just to give a last two slides, just to give you the glimpse that it is not dealt with the stem cells. That stem cell is also important in place of the IVF professions. It is important that how it comes into the potential of the different types of stem cells in the medicine, including the IVF, that it is the different stages you are very much aware of. Up to the 8 cell stages, they can be differentiated into individual, independent individual, which is not possible in the human, but in case of the dogs and cats, that is possible. That's why you have 3, 4, or 5, 6 the kittens or the babies. But then, and the 2-cell stage, you can have at least 2 or 3 cells. The 4-cell stage, you have the tetrad also, and the production of the babies. And 2-cell stage, very common, that is the identical twins or fraternal twins. So this is a picture of the twins, and this is a meeting held in the US. These are our twins, identical twins. So these stem cells are the specific type of undifferentiated mother cells of the body with a capacity to unlimited prolonged self-renewal, not that dividing self-renewal. When there is a need, they can replace the cells and have the ability to differentiate into different range of specialized cells, including the gametes, the ovum and sperms. So that is the beauty. These are the 2-person Gordon and Monica in 2013, 12. They got the Nobel Prize for the physiology and medicine. They discovered this, and that they discovered the iPSC, induced pluripotent stem cell, creating induced pluripotent stem cell. What does it mean? That has made a world of difference. You get the stem cells from the baby, the embryo, or any other places. on the bone marrow. But here you can generate the stem cells in this culture, in the laboratory. That is, you take the diploid cells from the skin or any part of the body, from the male or female, and then that can be the packaged cells, the fibroblast can be packaged cells into the virus, and these cells can be used to develop you put the genes, particular genes, the nano KLF4 and OCT4 and SOX2 inhibitory these, and this virus in a package in the virus, and this virus is modified with the 4 different genes, and then these genes and these genes virus is mixed with the cells, and the cells then that transformed into, or they in the 8 to 9 days or 10 days time, and the post-infection, they in fact we generally packaged in the virus, and this virus is modified with the all these 4 types of genes or the transcription factors called, and then the cells are infected with this virus, and 8 to 9 days time, this post-infection, that the colonies of the cells will, similar properties of the embryonic stem cells can be identified. So these cells can be differentiated the way you want. If you want to differentiate into the gametes, the ova or sperm, that is possible. That's why the male and female, those who are same-sex marriages, they can have their babies now. And the IVF people can think of giving them a joy of having their own child, carrying their own genes, they having the male and female couples. So otherwise also can be artificially, can be not artificially, it is a live cells, can be made the gametes. So these are I wanted to tell something
Sir,
of this. So
sir, I'm really sorry, sir.
So this is the pattern you can do with the stem cell breakthrough, and the lab made babies possible. And this is, I come to the last. So this can be the use of potential in the including the stem cell profession. It can be the IVF profession can be used. And these are my earlier slides. So thanks very much. I'm sorry I have taken a little more time. Sorry to about this. But nevertheless, thanks for the opportunity. And if you have any questions, you can ask when the question session comes. I will be there. Thank you.
Thank
very
you,
much.
sir. Thank you, sir. It was really, really wonderful knowing all the, briefly starting from nomenclature to the future use. It was a very interesting talk, and we would really love to hear from you, and hoping to listen to you in the question answer session.